Hi, I'm Teresa Zagoda, and here are some of the images I have captured throughout college and work as a microscopy technician. These crystal melts were made dissolving hippuric acid crystals or granulated urea in isopropyl alcohol, then placing the solution on a slide. Holding the slide over a hot plate sped up the evaporation of the acid solution, allowing crystals to form. I photographed the crystals using cross-polarization and a quartz wedge to bring out colors not visible to the naked eye. These are some of my favorite images to make because they produce such beautiful colors and shapes that almost look like a painting. The hippuric acid and urea in these images are biological waste products found in urine, and the isopropyl alcohol is the main ingredient in drugstore rubbing alcohol. This snowflake portrait was taken with a microscope and slides that had been left outside to acclimate to the cold, so that the snowflake wouldn't melt immediately as soon as I put it on the slide. I added color by placing gels over the light source. I found this skeleton shrimp in an ocean water sample collected near the Marine Biological Laboratory in Woodsville, Massachusetts. It displays autofluorescence, biological molecules glowing in response to certain wavelengths of laser stimulation. This diaphanized skate was photographed using bright field illumination, which yields a completely different image than the fluorescent version you see in my article. In this confocal image of pine pollen, biochemical differences between the outer shell and the inner portion of the pollen grain shows up as two distinct colors of fluorescence. False color highlights the autofluorescence in this confocal image of Taenia solium, better known as a pork tapeworm. I chose to orient this image in this way because I thought it looked like the face of some alien creature. This is a confocal image of an Obelia hydroid, a carnivorous marine invertebrate distantly related to jellyfish. This little animal is commonly found in coastal waters around the world. Read more about my work and see more of my images in my article, A Journey Through the Microscope, in this special issue of American Scientist.